Hi everyone, this is Théotime Grohens. I'm a PhD student at INSA Lyon under the supervision of Guillaume Beslon, and I'm really happy to be here today presenting to you my work jointly with Meyer and Guillaume on a genome-wide evolutionary simulation of the transcription-supercoiling coupling. So what is DNA supercoiling? The DNA molecule is made up of two strands that rotate around each other in a relaxed state. They take around 10.5 base pairs to turn around each other, but they can also twist and riff more or less because the DNA molecule is very flexible. When DNA is overwound, the typical length of a turn is less than 10.5 base pairs, and when DNA is underwound, the typical length of a turn is more than 10.5 base pairs. And this phenomenon is called supercoiling, which we will call sigma. So supercoiling represents the density of extra twists per base pair along the genome. So when the DNA is overwound, supercoiling is positive, and when the DNA is underwound, supercoiling is negative. On top of this, DNA can also riff around itself, so make those sort of loops. We're not going to be talking about that today. And in bacteria, which we're going to focus on today, the DNA molecule is usually slightly relaxed, which means that sigma is negative. So DNA supercoiling is tightly regulated in bacteria, mostly by a class of enzymes called topoisomerases. And the two main ones are gyrase, which decreases sigma when it's too high, as we can see here on the right-hand side of this panel. When supercoiling is high, gyrase activity is high and lowers supercoiling. And topoisomerase 1, which has the opposite effect. So when sigma is very negative, or we have very high negative supercoiling, topoisomerase is very active and tends to return DNA to its relaxed state. On top of this, structural proteins, which are called NAPs, create topological domains with different supercoiling levels, which we can see here in this lower panel, which represents the level of supercoiling along the genome of an E. coli bacteria, so which changes along the genome. And on top of this, supercoiling also varies during bacterial life cycle and as a response to environmental variations. So supercoiling is a very dynamic property of the genome, but it's also very tightly regulated. And it happens that DNA supercoiling has an effect on gene transcription. So how so? When a gene starts to be transcribed, this is because an RNA polymerase molecule has attached to the DNA and starts transcribing. And for this RNA polymerase to attach, it needs to pry open the DNA molecule. And this actually is easier the more underground the DNA is, so the higher level of negative supercoiling we have. And so this is reflected in those two graphs here. The top part showing the negative energy needed to open the DNA, which increases as supercoiling gets more and more negative, and the lower part showing the relative expression level of a gene on the plasmid depending on the supercoiling level. And we can also see that the higher the level of negative supercoiling, the higher the gene expression level. So DNA supercoiling has an effect on transcription, but transcription also has an effect on DNA supercoiling. So this figure comes from a classical paper by Liu and Wang in 1987. And the main idea is that the transcription complex, so made out of the RNA polymerase itself, the RNA that's being transcribed, and also the ribosome, which is translating this RNA into a gene, into a protein, is very bulky, and so it cannot easily rotate along the transcribed RNA. And so this generates a buildup in twists downstream of the transcribed gene, and a lack of twists upstream of the transcribed gene, which corresponds to a buildup in positive supercoiling downstream and negative supercoiling upstream of the gene. The supercoiling also changes due to gene transcription. And so if we put all of this together, we see that there is a coupling between transcription and supercoiling, which is due to this back and forth effect between transcription and supercoiling. And so if we have a gene A, which is being transcribed, then it's going to change the level of supercoiling around itself. And so if a gene B is in the neighborhood of gene A, its expression level will change, and this change in the expression level of gene B will in turn affect supercoiling at gene A into the expression level of gene A. And so depending on the two relative orientations of genes A and B, we have three possibilities. Either genes A and B are in diverging orientations, in which case they both positively reinforce each other's expression because they are both upstream of each other, or when they are in converging orientations, they, on the other hand, inhibit each other. And finally, if they're in parallel orientations, then one of them is going to activate the other one, and the other one is going to inhibit the first one. So depending on those relative orientations, behaviors can happen due to this coupling. And so sort of the question I wanted to ask here that hasn't really been asked before is what happens at the genome scale, because what I've just described is local interactions between genes. But in bacteria such as E. coli, genes are on average 1,000 base pairs apart. And this happens to also be sort of the typical distance at which those supercoiling effects due to transcription are being felt. And so it could be theoretically possible that, well, each gene interacting with their neighbor causes a genome-wide network to appear instead of just local islands of interacting genes. And in particular, in such bacteria as Dickia tadentia, different set of genes are activated in response to environmental changes, which change the level of supercoiling. And so we are also interested in seeing whether this response in the change in gene expression levels could be mediated by the transcription-supercoiling coupling. So the way we tackled this question is we created a model as simple as possible to look at the transcription-supercoiling coupling. So it's being described here. We have individuals that have a circular genome in which we represent genes using a string of pairs. So that means that each gene is a dot on this genome with some level of intergenic distance between neighboring genes. And at each time step of an individual's life cycle, we're going to update the expression level of each gene, depending on the expression levels of the nearby genes through the transcription-supercoiling coupling. So the outer ring is the positions of each gene on the genome, and the inner ring here represents the interaction network between neighboring genes. And finally, we're going to add some environmental conditions that are going to influence gene expression levels to see how they react to these different conditions. So mathematically, the way we formulated this is that for each gene, we compute the local variation in supercoiling. So at gene I, we look at all the interactions with all other genes J, which is, to be as simple as possible, just a linearly decreasing with the distance until a certain threshold. And then to get the level of supercoiling at this gene, we're just going to sum up all these interactions times the level of expression of that gene. And finally, we're going to feed all of this to an activation function that's going to take into account the level of supercoiling at the gene and the environmental conditions to give us the expression level of the gene at the next time step. So what does this actually look like in practice? So this is an example individual on the left-hand side and its interaction network. And on the right-hand side is the gene activation levels over time, or the gene expression levels over time. And we see that, for example, genes A and B here are in divergent positions and so should be activating each other. And this is actually what we can see in their expression levels. So A is in deep purple and B is in deep red, and we can see that they both converge to an activated state. Whereas, for example, genes C and D are being repressed by each other. And so C here is in pink. So C is being repressed both by B and D. And D itself is being activated by E, and so D actually ends up activated. So how do the environmental conditions influence our gene network? So the idea is that we introduce parameter beta that represents a variation in the basal supercoiling level compared to a baseline. So beta equals sigma minus sigma 0 over sigma 0. And the higher level of beta means a higher level of negative sigma of supercoiling. So it means that it's easier for genes to be activated and a lower beta means that it's harder for genes to be activated. And so on the left-hand side here, I'm showing the gene expression levels over time of the same individual, but in different environments, so for different values of beta. And we see that as beta gets progressively higher, more and more genes end up being activated because it's easier and easier for genes to be activated in this environment. We also see that some genes keep on being repressed because of their interactions with their neighbors. So all in all, we are sort of happy with this model because it shows local activation and inhibition of neighboring genes due to the supercoiling and transcription coupling. It shows that we have genome-wide interaction networks that can emerge from those local interactions. Gene expression levels do change as a response to environmental conditions. So it sort of has all of the ingredients that we wanted. I know the real question I wanted to ask is, well, how do these gene networks perform under evolution, and do they allow for adaptation to different environments? So the way that we set this up is that we chose to evaluate individuals in two different environments, so which we call A and B, which are characterized by different values of beta. And so on the left-hand side here, I show the same individual being evaluated in the two environments. So we see that the gene expression levels differ. And for this individual, we decide to split genes into three classes, A, B, and AB. And basically, the evolutionary goal that individuals should evolve towards is that genes of class A should be activated in environment A, but not B. Genes of class B should be activated in environment B, but not A, and genes of class AB should be activated in both environments. And so in order to have evolution, we need to have a mutational operator, and which is the simplest one, which is genomic inversions. So the idea is that in order to make an inversion, we pick 2 points at random in the genome. So for example, here, P1 and P2 on the individual that I presented before. And we simply reverse the part of the genome that's between those 2 points. So we change the gene orientations and the order along the genome. And as we can see here, so this is the phenotype before the inversion, after the inversion. For example, gene B that used to be activated here because it was interacting with gene A before the mutation is actually inhibited after because it's being repressed by gene B. So those inversions actually change the phenotype of the individuals. And so with all those ingredients in hand, what we did is that we evolved 15 populations of 100 individuals for 200,000 generations, each time starting from clones of an individual with 60 genes, so 20 of each class, that were equally distributed along the genome that is 60,000 base pair long with random orientations. And at each generation, we evaluate all individuals in both environments and compute a fitness level based on this. We select the producers according to their fitness using a fitness proportional distribution, and then we make those reproduce and randomly undergo mutations. So this is the result. On the left-hand side is the gene activation levels. So the proportion of activated genes of each class in each environment, so A on top and B on bottom, for the best individual at each generation of replicate 3, the sustainable replicates. So for AB genes in blue, we see that they're well expressed in both environments as required by the evolutionary target. For B genes in green, we also see that they learn to be activated in environment B here and repressed in environment A here. Also, basically all of them. And for A genes in red, we see that they also display the same tendency of being activated in environment A and repressed in environment B, even though the tendency is less strong. There's also caused by the model in which it's easier to activate in B, which is a higher level of beta than A in area cell. And finally, we can see that fitness here in light blue is still increasing at the end of simulations. So adaptation is still taking place, and probably those expression levels could still evolve. So if we average over all of the populations, we see that there is a clear signal for AB genes to be activated all the time, for A genes to be activated in the environment A but not B, and even more clearly for B genes to be activated in environment B but not A. And so for each class of genes, there is an adaptive response to each environmental condition that has evolved. So finally, as a conclusion, in our evolutionary model, we saw that a well-differentiated gene expression level can repeatedly evolve as a response to the set of two different environmental conditions for each class of genes. And the transcription-supercoiling coupling, along with those genomic inversions, is sufficient to generate this pattern of differential activation and inhibition of classes of genes. So that's really nice. And we believe that this suggests that the transcription-supercoiling coupling is a plausible mechanism to explain the environment-dependent gene expression patterns that we can see in bacteria. And as future work, we would be interested in, we're working on looking further into the structure of those genomes that have evolved. And I've shown here what a typical genome of an individual looks like after the end of the simulation, so 200,000 generations, and the gene interaction network that's associated. So that's not in the paper at ALIFE, but we hope to publish that soon. And with that, I would like to thank you for listening to the talk, and I'll be glad to take any questions. Thank you.